Today we are going to discuss the chromosomal aberration, in which we will discuss duplication. There will be the different types of duplication, and there are various aspects of the chromosomal duplication, that is the meiotic pairing, and there are various types. So we will proceed with this particular topic. The another type of chromosomal aberration is duplication, or you can say the addition. Duplication is the presence of an additional chromosome segment as compared to the normally present. It is of four types. First one is the tandem duplication, another is reverse tandem, third is displaced duplication, and the fourth one is transposition. Now come to the first one. This is the general introduction of the tandem and reverse tandem. We will discuss it with the help of the photograph. You just find out that this is the normal sequence. You just see that A, B, C, D, E, F, G, H, I, J are the genes placed, and this is the circle which is presenting the centromere. You just find out that we are having this the fragment, or you can say the segment, that is DE. This is the segment which is to be duplicated in the sequence which is given below. The first we are saying the tandem duplication. Just find out what is tandem duplication. We are having the normal sequence A, B, C, D, E. Now this is the darker portion which you are finding. This is again added, and this is called as, such type of duplication is called as tandem duplication. You just find out that we are having A, B, C and D and E. So DE and again DE, the presence of DE is showing that is the addition of the segment. Now there is the another case which is called as reverse tandem. In reverse tandem, you just find out that the normal sequence is A, B, C, D, E, and this ED segment is again added in a reverse order. So it become A, B, C, D, E and ED again, and we are having the normal chromosome once again, the normal sequence. So ED portion is added in the reverse order. This is called as reverse tandem. Now there is the displaced duplication. In this particular picture, the displaced duplication is again divided into two aspects. One is the one which is called as homobranchial, that is on the same arm. How it will happen? We are having as come to the first figure, A, B, C, D, E. This is the normal sequence. Now what is the condition? After A, the DE portion, that is, this is the darker portion which we have earlier, this portion get added, and again we are having B, C, D, E, F, G, H, I, J. This is called as displaced duplication, which is homobranchial in nature, means the addition of the genes is taking place on the same arm. Now there is the another one that is called as heterobranchial. In heterobranchial duplication, we are having the addition of the gene on the different arm. You just find out that this particular arm remains the same, and after FGH again DE added, and it will remain IJ as such. So this is called as the heterobranchial duplication, that is the displaced duplication. The next one is called as the transposition. Transposition is where we are having the addition of the genes on the non-homolog, that is we are having the first one A, B, C, D, E, F, G, H, I, J, and on the non-homolog we are having K, L, M, N, O, P, Q, and here after Q, D, E is added, and after that it is R, S, T. So this is called as transposition. We are having the extra chromosome. This D is showing the presence of the extra chromosome. This is the type of the duplication. There are four different types of duplication we just discussed here. Now how the duplication is produced. Now the chromosome duplication is usually caused by abnormal event during recombination. This is the figure which is showing the duplication, how it will happen. This is misaligned crossover. How it will happen? we are having the sequence of the gene A, B, C, D. And here we are again having A, B, C, D. There is the crossing over taking place. Here we are having between CD and here we are having BC. What will happen when this misaligned crossing over is happening? This is the first chromosome, which is the normal one, A, B, C, D. You just find out. Now, in the next case, what will happen? We are having the duplication of the chromosome. It will proceed in this manner. It is like this: A, B, C. Then this C is here. Again, it come down and we are having C, D. So, you just find out A, B, C, C, D. So, there is the duplication of the gene C occurs. There is the another case in which we are having A, B and it comes here, it is D. So what is the condition? In this particular case, there is a deletion of the gene C occurs. So we are saying that the chromosomal duplication is usually caused by abnormal event which is during recombination, and in which we are having duplication as well as the deletion of the genes. There is the another interesting aspect in the case of duplication, that is called as meiotic pairing. Now you just find out this is the normal chromosome and this is the another case in which we are having the chromosomal duplication. The sequence of the gene is A, B, C, D, E, F, E, F, G. You just find out that in this particular case, that E, F gene, which is shown with the blue and purple color, is duplicated again. We are having E, F genes here and again E, F genes are added. Now, in case of alignment in prophase 1 of meiosis, what we can find out that this is the normal chromosome. The another chromosome, which is the duplicated one, is forming a loop. Why it is forming a loop? Because this particular duplicated chromosome is tried to align with the normal chromosome. So the duplicated portion is forming a loop. So you just find out the duplicated EF region must loop out to allow the homologous sequence of the chromosome to align. So in case of you just keep in mind that in case of deficiency as well as in case of duplication, that particular abnormal chromosome is forming. In case of deficiency, the normal chromosome is forming the loop, and in case of duplication, the duplicated chromosome is forming the loop here. Now, there is one of the very interesting example of the duplication. It is the bar eye phenotype in Drosophila. This particular condition is that in which there is the reduced number of ommatidia. You can find out the eye facets get reduced. There is the condition which is called as Ultrabar. In Ultrabar, you can find out that we are having a very few number of facets than the bar homozygote. Now, what is the condition? This is the normal Drosophila in which the number of eye facets are 810. When one of the segments, 16A segment, get introduced in that particular case, there is the bar heterozygote get formed and we are having 350 facets. The number of the eye facets get reduced, and in case of bar homozygote it becomes 70, and in case of ultra bar homozygote condition we are having the 45 eye facets and it reduces the number of the eye facets. We will, in the next lecture, we will discuss what is the types of the duplication in reference to the addition of the 16A segment. This is the ultrabar condition which we are discussing. This is the homozygote, and you can find out in homozygote the number get reduced, it become 25. In heterozygote it is much higher, that is the 45. Thank you.